Bismillahirrahmanirrahim, and this is the final revision. We have your exam tomorrow morning, inshallah. And I wish you all the very best. And these are some recently some topics which I just want to quickly revise with you so that you are ready for the exam tomorrow. Now, the first and the foremost thing to remember is that you have done nothing. You might have prepared very well, but at the time of the exam, you might just freak out. So it's all Allah's blessings that you have, which will help you to ace this exam. So number one, I ask you all to offer your Fajr prayer. So put the alarm. You might have slept late or whatever, but offer Fajr. Then pray to Allah for guidance and knowledge. Number three, I've asked you all to have some sweet things for breakfast. You might not want to have an egg or something, but you should have some chocolates or ice cream. Why? Because the brain can only use glucose. It's the brain is the respiratory fuel is glucose. So if you are high on glucose, you are going to be able to really function well in the exam. It's only 1 hour 45-minute exam. So it's not going to be such a long exam. Then if some of you need something to calm your nerves, you can have some chewing gum. And if you haven't slept or you have not been very well the day before, then you can take a Panadol. You can take, depending if it works on you, one tablet. On me, I can't work on one tablet. I usually have two tablets. So this is the brief guidance to you, but basically you have to ask Allah for the guidance and the knowledge which you have in answering the questions in the exam. The most important point in the new syllabus is the use of a hydrogen carbonate indicator. Now, where you're going to go wrong is if you have not understood this, what it is used for. What is this used for? It is used for a type of pH indicator that is sensitive enough to show a color change as the concentration of carbon dioxide gas in an aqueous solution increases. So if the carbon dioxide is high, it's yellow. It's okay, it's medium. If the carbon dioxide is low, produces a weak acid solution. So you see, carbon dioxide dissolves in the water to form carbonic acid. So more the carbon dioxide, more the carbonic acid. Less the carbon dioxide, less the carbonic acid. So hydrogen carbonate indicator responds to changing CO2 levels in the environment. This can be used to see whether or not plant requires carbon dioxide for photosynthesis. So CO2 levels decrease. That means what is happening? That means photosynthesis is taking place. The carbon dioxide is being used up for photosynthesis. If you look at it, when will there be high carbon dioxide? When there will be high carbon dioxide when respiration is more than photosynthesis. Respiration produces carbon dioxide, photosynthesis uses carbon dioxide. So there'll be more carbon dioxide when there is more respiration. So the color is going to be yellow. More carbon dioxide means more carbonic acid. This is a pH indicator. But if photosynthesis is more, that means there's going to be less carbon dioxide. Why? Because the carbon dioxide is being used up for photosynthesis. In this room, say there's 100 molecules of carbon dioxide and I have a plant here. So the carbon dioxide is going to be used up by the plant. So there'll be low carbon dioxide when there is photosynthesis and the color will turn to purple. So this is a hydrogen carbonate indicator. How would you study the rate of photosynthesis? Or how would you study it when rate of photosynthesis and respiration is the same or it is more? So in bright light, you see what is happening. In bright light, there is more photosynthesis. So the hydrogen carbonate indicator becomes red. Why? More carbon dioxide. That means the carbon dioxide is being taken up by photosynthesis. So there's less carbon dioxide in the air. But when you have covered this plant with a dark card or an aluminium foil, what is happening? There's going to be more carbon dioxide because the plant is respiring now only. Previously it was respiring, but then it was photosynthesizing. So in this situation, photosynthesis was more than respiration. In this situation, there is no photosynthesis because there's a dark card around it, so there's no light entering it. And this is the control. Control means nothing in it, and still if there's a color change, then it would mean it's not the leaves, but there's just a color change because of something in the air. The control is for comparison. Now, what you have to realize is what are you doing a tube A in the dark, tube B in the light, tube C in the control. Important thing is equal volumes of the bicarbonate indicator. And what are the variables? The different variables in the experiment illustrate. So, what are the leaf size, volume of the indicator starting color, the volume of air in the test tube? These are the variables that you need to control and need to be the same. And how are you measuring the rate of photosynthesis? By the CO2 change. More the rate of photosynthesis, more the CO2 will be taken up. More the rate of photosynthesis means more the CO2 will be taken up. Now, this is an experiment showing the formation of carbon dioxide by respiration. So you have germinating soaked bean seeds, you have gauze or cotton wool, and you have an hydrogen. So color change is yellow. Why is it yellow? Because there's more CO2. But here, there's no color change. Why? Because we had dead boiled beans or peas. And then we would have washed them in some sort of a sterilizer, some Dettol or something. Why? Because they would be decomposers on it. If you just use boiled seeds, it will have bacteria and fungi on it. But if you have washed them in bleach or chlorine, then of course they won't have any of the bacteria and fungi. Now, why I'm showing you this diagram, the reason is that I fear that you might mix something up with this. There's the hydrogen carbonate indicator solution. But then what is this? What is this? Do you all remember that? You see, what we did is we added the water with sodium hydrogen carbonate. Now, this is water with sodium hydrogen carbonate. Now, don't mix this with that hydrogen carbonate indicator. Here we added sodium hydrogen carbonate to provide the CO2 because this is an aquatic plant. This is an aquatic plant. So an aquatic plant was probably in a pond, and in a pond there would be snails and there would be frogs and there would be tadpoles which would be respiring and producing carbon dioxide, or there could be fish. Now here we've taken the pond weed and we are doing this experiment in the lab. So we're using tap water. To that tap water, we have provided a chemical which is going to produce carbon dioxide. I don't want you mixing up this with that hydrogen carbonate indicator solution. That's what my worry is. Now in this experiment, you can see factor being investigated is the distance of the lamp. So that is the independent variable. Then there's a glass tank filled with water. Why? That's the heat shield. There's a lamp. Then there's a thermometer to monitor temperature. That's a control variable, CV. Then there's inverted boiling tube where the oxygen is going to collect and then we can measure the volume. Then this is the water, so the volume must be the same if we're going to control this variable. And then if we are going to change the sodium hydrogen carbonate, if we might use a 1% solution, next time we use a 2% solution, next time we use a 3% solution. Why? Because we are investigating how carbon dioxide concentration will affect the rate of photosynthesis. If we are measuring light, then we will not change anything else. We'll only move the lamp. If we are measuring how CO2 concentration will affect the rate of photosynthesis, then the lamp will stay here. Then we won't move the lamp. Then we won't move the lamp, everything will stay here. The pond weed must remain the same. So we're investigating light intensity, CO2 concentration and temperature. If we are going to change the temperature, then we're going to place this in a water bath. Then we're going to place this in a water bath at 3 or 4 or 5 different temperatures, 30, 35, 40, 45, 50 degrees Celsius. So my worry is that this is sodium hydrogen carbonate solution we've added to provide it the carbon dioxide. Do not confuse this with the hydrogen carbonate indicator solution which I've just talked about. Hydrogen carbonate indicator solution responds to changing CO2 levels in the environment. This can be used whether or not a plant requires CO2 for photosynthesis, or if we can compare respiration and photosynthesis. Now coming to another point, where has this section been taken up? Where are we seeing this diagram? You can see it here. The section has been taken through the heart here, and then we are looking at it from the top. So you know this is the bicuspid valve because there are two flaps here. This is the tricuspid valve because there are three flaps here, and this is the aorta and the pulmonary artery. And the semilunar valves are also three flaps. So this identification, I don't know, I have a feeling they might give you some sort of a question like that. My feeling could be wrong, my feeling could be correct. Please do not think that I know the paper. I'm just giving you what my gut feelings are. Another diagram showing you the transverse section. Just makes it very interesting, makes it looking from the top, how it's going to look like. You see, I have the syllabus in front of me and I am looking at things which could possibly come in the exam, and it's not a very easy situation for me. I am just making wild guesses to help you all. And in the syllabus, there is this point. It says identify on images. The syllabus says identify on diagrams and images the ribs, internal and external intercostal muscles, and the diaphragm. Then it says identify on diagrams and images the larynx, trachea, lungs, bronchi, bronchioles, alveoli, and associated capillaries. So I'm going on the identify. The few identify I feel are important ones. Now you can see here the ribs. But what we have to identify is the external intercostal muscles and the internal intercostal muscles. Now I've made it a little easier for you is I've made a V, and this is the external and these are the internal, and you can see the internals are something like this. So this is the internal ones and this is the external intercostal muscles, and external intercostal muscles contract to move the rib cage upwards and outwards to increase the volume of the chest cavity, so that then there's a pressure difference created which causes air to move in by high pressure to low pressure. Another diagram showing you the external intercostal muscles and the internal intercostal muscles. So they can give you a number of diagrams and you have to be very clear on it. What is the sort of way to identify this? Syllabus says you have to identify larynx. Then you have to identify trachea. Then the right and the left bronchus, the bronchi and the bronchioles and the alveoli. So you must be able to do these diagrams. You must be able to identify larynx, trachea, bronchi, bronchioles, and alveoli. So you must look at a few diagrams. I'm sure you've done that. There is this diagram as well. You can see the larynx here, trachea, bronchi, alveoli, bronchioles. Then coming to a new topic, active immunity and passive immunity. Active immunity, I always say, is when your lymphocytes perform some activity. So if you have an infection like you had measles and you were sick for 10 days, you did not go to school, so you had an infection, the pathogen entered your body, measles is caused by a virus, and you were sick for 10 days, and then after that, you were fine for the rest of your life. You never had measles again. That's natural active immunity. But if I give you vaccination, like polio vaccine in the form of oral drops, well, we give you weakened pathogen. And again, your lymphocytes think they're the real ones and the real bad guys, and they make antibodies against it and they develop memory cells. Now, that will be artificial active immunity. Why is it artificial active? Because your lymphocytes did the activity. So, in active immunity, it's your lymphocytes, whether it happens because of an infection or whether it happens because of a vaccination in which we give you the weakened pathogen. Please do not use the word germs. Germs is not a word we are allowed here in biology. Now, in passive immunity, what we give you is we give you antibodies. So, if it is a natural process like mother to fetus, and these are called the maternal antibodies, either in breast milk or across the placenta. Now, that will be called natural passive immunity. So, we're not giving you the pathogen, we're giving you the ready-made antibodies. And if it is an artificial, then it is when we made the antibodies in the lab and we are injecting those into you like we do it if you've had a injury on the road. So we think you might have some tetanus bacteria entering your body and that they are not very good. They cause a very lethal disease. Person can die of tetanus. So we give you an injection and you go to the hospital and you say, I just fell on the road and I want a tetanus injection. But that's for a short time. The antibodies that we give you will only work for 6 months to a year. And then you'll have to have the injection again. So they are short living. Just a quick Venn diagram. Active makes own antibodies, exposed to pathogens, defense change to fight pathogen, long lasting. Example includes vaccines as well. So active immunity. Then passive immunity is given antibodies, is not exposed to the pathogen. In passive immunity, you are not exposed to the pathogen. The mother's ready-made antibodies enter the fetus. Defenses don't change, short lasting. Example, mother to child. Now, what is the common thing? Protect body from infection, and both have natural and artificial variations. Then coming to differences between arteries, veins, and capillaries. Arteries have thick wall, they have more elastic tissue, and they have muscles. Now, these muscles are smooth muscles in the wall of the arteries, and these contract and relax. That's when we talk about vasoconstriction and vasodilation. And then, of course, the artery forms the arteriole. So you've got the artery here, then the arteriole, and then we have the capillaries. Capillaries are one cell thick. They allow substances to move in and out. So the function of the transport system occurs actually in the capillaries. If you say oxygen is transported, yes, it's transported, but it moves out of the blood into the cells only in the capillaries. It cannot move out of arteries and veins. They are thick walls. Veins carry blood back to the heart. They are thin walled. They have a little bit of elastic tissue, and they have very little muscular muscles in them as well. But the major arteries have more elastic tissue. Why arteries? Because arteries arise from the aorta. Aorta arises from the left ventricle, and the wall of the left ventricle is thicker, so exerts a lot of force. So the aorta carries blood at a very high pressure. So all the arteries are arising from the aorta. So the pressure is actually being generated by the wall of the left ventricle. These are micrographs showing you the artery, capillary, and the vein. You should be able to differentiate between them, and you should be able to look at these diagrams and tell me which is an artery and which is a vein and which is a capillary. So you're supposed to be looking at photomicrographs and the images of artery, veins, and capillaries. Looking at a diagram of the kidney, what do we have here? We have the renal artery and the renal vein. Now, everybody has to be clear about it. Renal artery is a branch of the aorta and is carrying blood at high pressure. So renal artery carrying blood into the and renal vein carrying it out and will enter into the vena cava. Similarly, the renal artery is coming from the aorta and is arising from the aorta, and so we have got to understand that. Then it goes in, and then the outer part of the kidney is called the cortex, the inner is called the medulla, and the inner part is called the renal pelvis. So quick recap, the adrenal gland is placed on top of the kidney. There are the two kidneys, there's the renal artery, renal vein, then these are the ureters. So two ureters, right and left ureter. Then there's the urinary bladder, and then there is the urethra. So all these you have got to know. The labeling you must know. Aorta and vena cava, favorite ones that they ask you, keep on asking you. For the new syllabus, you've got to realize is they want more details. They want you to know what is the glomerulus. So there is a glomerulus, and there are the glomerular capillaries. So this is the glomerulus here, which we are going to be looking at here. Then there is the Bowman's capsule. Then there is the proximal convoluted tubule. And then there is the distal convoluted tubule, which is this one. And then there is the collecting duct. Another diagram explaining this to you, glomerulus, closely packed group of capillaries. And it is coming, there's a branch of it coming from the renal artery, which is called the afferent arteriole, and then the one leaving it is called the efferent arteriole. Then the first coiled tube, which is also called the proximal convoluted tubule, you don't have to know the details of it. Then this is the loop of Henle, which we all know this U-shaped structure right here, down here, which we goes here. This is the loop of Henle. It's in the syllabus. And this is the second convoluted tubule. And then this empties into a tube, which is called the collecting duct. So what is going to happen here is filtration, ultrafiltration. And then in this part, there is going to be selective reabsorption. There is going to be selective reabsorption. You see, everything is going to filter out, but then we don't want the useful stuff to be lost in the urine. The urine is only going to contain urea, excess water, and excess salts. That's it. The urine is going to contain only three things. Urea, you can't say water, it's excess water, and excess ions. So urine contains urea, excess water, and excess ions. So what is going to happen here is in the glomerulus, we're going to have ultrafiltration. And then in this part, in the rest of the part, we're going to have selective reabsorption. Selective reabsorption means only glucose, amino acids, things which are useful to the body have to be retained. So they must go back into the blood. That is why you're having this blood supply here. is going to back enter into the blood. Only what we want to remove is the urea and the excess water and the excess salts. Another diagram showing you the glomerulus and the Bowman's capsule, and you can see here filtration is occurring here. And then as these things filter out and go here, then they're going to be reabsorbed into the blood. The blood is not going to reabsorb it. They're going to be reabsorbed and the blood is just going to carry it away. This is another favorite question of the examiners. What is the role of the blood capillaries? The role of the blood capillaries is just to carry away the dissolved substances, to carry away the reabsorbed substances. If you throw something into the river is just going to carry it away. You are throwing something into the river, so actually reabsorption takes place and the blood is only going to carry away the reabsorbed substances. Very good explanation. Small molecules such as water, glucose, salts and urea are squeezed out of the blood into the renal capsule. Reabsorption, any useful substances such as water and glucose are taken back into the blood. The remaining liquid called urine flows into the ureter. Now, as you look at the new syllabus says describe the structure of a synapse, including the presence of vesicles containing neurotransmitter molecules, the synaptic gap, and receptor proteins. So you have to know this. It's the syllabus point, structure of a synapse. So there is a presynaptic neuron, and then there is a postsynaptic neuron. So this would be the presynaptic neuron, and this gap in between is called the synaptic gap. There's a microscopic gap. Now, what happens? Then you have to know there are synaptic vesicles, then there are mitochondrion, because a lot of energy is needed here for production of these vesicles and other processes. So an impulse arrives. So there's a nerve impulse arrives. What happens after the nerve impulse? The neurotransmitters are released. The vesicles move toward the synaptic gap and they fuse. And then they release these contents into this gap. And this gap, then these diffuse and they come and attach on the surface of these receptors. So these are the receptors. And this causes the sodium channels to open and sodium ions diffuse in. And an impulse is generated in the postsynaptic neuron. So again, another one example which I like this diagram very much. I think it's a beautiful diagram. Number one, this is the presynaptic neuron. This is the postsynaptic neuron. Then you have this gap in between, which is called the synaptic gap or the synaptic cleft. Then the neuron has a normal cytoplasm, and these are these calcium channels. So number one, nerve impulse arrives. Number two, calcium channels open. Calcium ions diffuse in. Number three, vesicles move and fuse with the presynaptic membrane. And then these neurotransmitters are released. And then these neurotransmitters will diffuse across the gap and they will bind to the receptors. And then this will cause an impulse to be generated in the postsynaptic neuron. This completes this video on most of the important topics and which I've discussed, and inshallah I hope all of your paper goes very well. Please remember to thank everybody. Showing gratitude is a very important trait of the Mormon Muslim, of the Mormon who knows that everything is because of Allah. Nothing is you. You are nothing. So thank your parents, thank your teachers, thank your home staff who ironed your uniform and you're wearing that uniform and going to school to take the exam. So thank everybody who's been there for you. Do not be think that everybody it's their duty for everybody to do something for you. Okay, thank you very much. Thank you for watching these videos and thank you for sending me all the feedback. And I wish you all the very best. If another video is needed, I will share it with you. I'll see if another new important topics are needed, then I'll send another video to you. Thank you very much.